Welcome back, guys. So we have talked previously about cell membranes and how they regulate what comes in and what comes out of a cell. So we are going to talk a bit about the natural processes that are kind of going on behind the scenes there. This should be review, and I'm not anticipating this being a very long video. It is probably going to be one of our shorter ones because a lot of this ought to be review from middle school. But we're going to go over it anyway, and I will show you your assignment that you'll be working on today. We are going to be kind of splitting this in half. I think it'd be a bit much to talk about tonicity and then also water potential, so just be on the lookout for a video with a whole lot of gnarly math, probably tomorrow. I'm sorry, guys, it is what it is. Okay. So to review, we have this tube, and the little dots represent some solute. It could be sugar, it could be salt, whatever it is, it clearly can't cross the little membrane in the middle of the U-bend. So we see in the first step, the water level is equal. It's just one side is clearly has more solute than the one on the left, okay? So since the solute can't travel, what's happening? So we see that the water itself is moving across the membrane, and this is osmosis. So remember that cells are going to want to be in equilibrium. All right, if you've heard that word before. So if it can't move the solute, it's going to move the water to make it more concentrated on that side, so that hopefully the amount of solute per gram of water can become the same on both sides of the cell membrane. So these are kind of like, this isn't something the cell's deciding to do, because as we're going to see, sometimes this can actually be lethal to the cell to do this. This is kind of a natural movement of particles sort of thing that is outside what the living thing is doing. Okay, so water balance. So this word I said before, tonicity, is the ability of extracellular, so extra means outside, and cell is a cell, so something outside the cell to cause a cell to gain or lose water. So depending on what kind of environment a cell is in, it can take water on or lose water, because the solution is going to want to reach an equilibrium where it's the same concentration on both sides. So depending on the concentration of, it depends on the concentration of solutes that cannot pass through the cell membrane. Because if they can pass through the cell membrane, instead of water moving, the solute will move, and it won't even be a deal. So cells can be in three types of solutions. We'll talk about all three, and you'll have to work with that on your assignment. So there's isotonic, hypertonic, and hypotonic. And we're going to stop and talk about that for just a second, because if you're taking notes, it'd probably be useful to have little definitions right there. So isotonic, like an isosceles triangle, it means that it is the same on both sides. Okay, an isosceles triangle, the two sides are the same length. The middle one doesn't have to be, like the bottom doesn't have to be the same length, but the two sides will be. So hypertonic, if you're hyperactive, you have too much energy. So it has more of the solute on the outside of the cell than on the inside. And hypotonic, if something's hypoallergenic, it has less of a substance that will cause allergies, so it has less solute if it's hypotonic. Okay, so take a second and get that sketched out. There's going to be more details on the following slides. I just wanted us to get this working definition as we move forward. Okay, so osmoregulation. We know about the word osmosis from middle school, so this is referring to regulating osmosis. So cells must be able to regulate their solute concentrations and maintain water balance. Cells are going to have ways to try to counteract osmosis if it's going to be negative, like those aquaporins we talked about with cell membranes. There's a reason why it says a lot of channels can be gated. We want to try to stave off some of this osmosis if it's bringing too much water in the cell or if it's making it shrink too much. Okay, so animal cells react differently than cells with cell walls. Cells with cell walls in a lot of ways have more protection against this than we do. Their little plastic bag is in a box, basically. That's what the cell wall does. So when you see the cells under a slide, you can see that there's kind of this little baggie with like chloroplast inside a clear box, basically, when you look at plant cells. And that little baggie can shrink or swell up, but it's hard for it to swell up so much it pops because it's got that protective box there. Anyway, cell walls, totally important. Okay. So cells immersed in an isotonic solution, so this is back to that ISO one, have no net movement of water. This does not mean that water doesn't move at all. This means that it is kind of drifting equally on both sides of the membrane. Let me move my face. Okay, the concentration of non-penetrating solutes is equal to that outside of the cell. So that is a fancy way of saying that sugar and salt that can't naturally get through the cell membrane is equal on both sides. Non-penetrating, we can figure that out. So water diffuses at the same rate, so the cell's gonna stay the same. That's kind of the root thing that I tell my biology 1 students. So the concentration is the same on both sides of the cell membrane. The cell's gonna stay the same. So when you go into the hospital, they don't give you pure water in your arm. They always ask for saline. What saline is, it's literally salt water. It's not super salty, but it's the same level of salty that your blood is, okay? And that is gonna be a super important thing. I may give y'all that case study as an extra assignment at some point. We'll just have to see. But people have died before because of mix-ups like this. So we want to keep our blood, like our blood plasma, isotonic to the red blood cells. Otherwise, bad news bears. Okay, so hypertonic solution. So if a cell's in hypertonic, it's going to lose water and shrink. And that's because a hypertonic solution has a higher concentration. So outside the cell, there's going to be more sugar or more salt or whatever. So water moves to the extracellular fluid. So it goes out of the cell. So a real world example of this, some of you might have killed slugs before by pouring salt on them. What you're doing is causing a highly hypertonic environment outside of that slug. Slugs don't have a dead layer of epidermis. So like on your hand feels dry because there's a layer of dead skin. That's actually important. It helps you to protect the living skin underneath. Otherwise, people would pour salt on you and you'd shrivel up. You don't want that. Yeah, so it's super important. That's why we regulate it. Like if you put your hand in a bag of Cheetos, your hand doesn't shrink up because it's so salty in there, okay? So yeah, osmoregulation at work. So a cell can shrivel and die, just like that slug shrivels and dies. And then there's this word plasmolysis. So the vacuole shrinks and the plasma membrane pulls away from the cell wall. This happens in plant cells. Okay, so usually it fills up the box nicely, but it can shrink a lot. And I may try to show you something from an assignment I gave Biology 1 because it's pretty cool and it shows this happening. All right, so hypotonic, they gain water. So the concentration is lower outside of the cell. So we're outside of the cell. So the cell gains water. So in animal cells, they swell and lyse is this word. Lyse means to pop. If we talk about immune system, there's other types of cells that lyse. But mostly we see this happening if you put an animal cell in a hypotonic solution, like pure water. So in these case studies, if someone gets pure water instead of saline, gradually their blood cells begin to pop. And obviously we know that form equals function, and if they're popped, they can't do their job, they're dead. So the person dies. They're not able to carry oxygen and they die kind of a slow, horrible death. So that's why it's so important. So animal cells actually work best under a hypotonic environment, which is weird, right? But because they need to maintain something called turgor pressure, they want to have a lot of water in their cells. It helps them the cell push against the cell wall, and this is what keeps plants upright. This is why if you put, say, like celery or an apple in the refrigerator for too long, the water's going to slowly evaporate out of that plant and it shrivels. It loses turgor pressure. Or if you don't water a plant and it gets super droopy, it's losing turgor pressure because you haven't given it enough pure water. So yeah, plant cells are kind of weird and they're definitely not like us. Okay, so that's it for the actual notes. Holy cow, Miss Vance, you got us done in 4 slides. What? Okay, so let's talk about some of this other stuff. All right, so y'all have a practice assignment. It's mostly osmosis, but y'all got the idea. We're going to need to be able to think about this going into water potential and the lab that we're going to be doing at the end of the week. Okay, so we got this cool little animated picture here. The little molecules are water, and the big green ones, you see the big green ones are non-permeable, meaning it can't go through there, right? So that's what we were working with in our assignment. So it says the graphic shows water molecules, small and blue, with larger green glucose molecules. So which side is hypertonic? Okay, so if the solute, the sugar, is green, I can count 1, 2, 3, 4 on side A, and, oh, it's moving, it's kind of hard. So there's like 10 or 11 over there on side B. So side B has to be the one that's hypertonic. So you can click on these charts in your assignment and you can type directly in here. So side B. So which side is going to gain water? So remember that water goes from high concentration to low concentration, just like a solute can. So the hypotonic side is the side that gains. Okay, so water is going to move from the hypotonic side to the hypertonic side. So if this were a cell, right, we could say that side B maybe is the inside of the cell and side A is outside of the cell. So since it's in the hypertonic cell is in a hypotonic solution, water is going to move toward side B. Okay, so side B, because it has so much more sugar, it's going to gain water to try to even it out. So side C, guys, we really ought to know what this is. That's the cell membrane. All right, so that's how you figure that out. If you're watching the video, you're getting all kind of help on this. All right, so this example, they are just drawing a cell for you and they are telling you in numbers how much sugar's on either side. So what percent of glucose is outside the cell? 80%. What side of water? So it has to add up to 100%, right? So if 80% of it is sugar, the percent of water would be 20%. So the amount of water inside the cell, so if it's 10% sugar, how much water is left? That's 90%. So is the water gonna move? We have 20% of water versus 90% of water. Is water gonna move? So yeah, osmosis is going to occur. So which way is it going to move, into or out of the cell? So I'm in a very hypertonic solution. So the cell is going to lose water. So it's going to move out of the cell. Is the cell going to shrink? Remember when we put a cell in a hypertonic solution, it shrinks. And we just said the cell is in a hypertonic solution. All right, so you have some other examples to work through. It looks like there's 3 problems like this one. And then you have a drag and drop here. So just like other drag and drops that we may have done, you click on an object like I did right here, and I can drag it to wherever it belongs. So this side has a lot more solute, so that's obviously my hypertonic solution. All right, so good luck with your assignment, guys. Let me know if you have any questions, and I will see you in the next video. Oh, quick reminder, at the end of this week, your campaign videos are going to be due. So make sure you get that done. All right, get with your groups, schedule how you want to take care of that. Don't leave it till the last minute. This is not the sort of assignment that you can do at the last minute. And because it's a video, it's going to be super obvious if you tried to get it from the internet. All right, it's going to be super obvious. So you can't really cheat your way out of this one. Either you do it and you get the points, or you don't do it and you don't get the points. And this is going to be your chance to talk to your voters, because remember, it's going to be open to all the Biology 1 sections to vote. So you got to be able to make your case to the rest of your class, right? All right, guys. So for real this time, good luck. See you in the next video. Please make sure you're getting your stuff done. Take care.